All right, so don't be shy. This is an interactive lecture. I would really love that during the lecture we interact with each other. So don't let me talk for hours and hours. Please interrupt me anytime you like. I really mean this. That's more fun for me, and it's more fun for you as well. Good. This, I think, is something that motivates me a lot. So if you read that quote, it's a very ancient quote by Plato, and it says basically, if you want to do science, you have to be amazed about the nature that's around you. You have to wonder, you have to be in awe about things that you see. And this is really the beginning of science. Science is not about, some math you construct for some ivory tower theory. It's really trying to understand natural phenomena as we see them. And that's the topic of this course. And what I want to do in the beginning is to give you some of those fascinating phenomena so you can see why you can wonder about all these different things. And once you have seen these movies and pictures, I'll move on and give you a more formal outline of the lecture, the material, the topics, the questions, the math, etc. So here you see quite an amazing movie. It's birds flocking in Rome at about this time of the year, I think. So what you see is that there's no leader, there's no particular bird that leads the crowd. It's really a self-organized phenomenon where all the birds together form these amazing structures on the sky of Rome. It's getting prettier and prettier as you look at it. By the way, people have put cameras on the roofs of Rome to actually measure the trajectories of these birds and infer how mathematical theory can actually account for the behavior, the self-organizing behavior of these flocks of birds. And that's one theme of this lecture. It doesn't need a person that organizes this, like here a picture of an orchestra which has somebody leading the orchestra. You don't need this in biological phenomena. And it's actually important that you don't need it, because otherwise it would be quite complicated to keep all things running. Here I show you some movies which are now at a much, much smaller scale. Before you saw birds, now you see single cells or single organisms. So what you see here first is a yeast cell, to be precise, budding yeast, so the stuff you make beer of. And what you see is that there is a pattern forming. So initially, the movie is looping here. You have a quite disorganized array of white spots, and then you have this polar cap that emerges here. And this polar cap is a set of proteins. And this set of proteins has a very particular role in the life cycle of a yeast cell. It forms a landmark. And this landmark here organizes structures that are indicated here by these rods. These are biopolymers, so-called actin filaments. They organize in bundles, and as they organize in bundles, they push out the yeast cell, so they create a bud, and this is then the origin of a new yeast cell, so the birth event of yeast. So you see this function to patterns. These patterns in biology are important for getting offsprings. This here is a quite nasty bug, E. coli. So if you have that too much in your stomach, that's not good. So these bacteria, they also have to divide at about actually a rate of 20 minutes. The way they do that is they have this oscillating pattern of proteins that goes back and forth between the two cell poles, and as it goes back and forth, between the two cell poles, it has a task. So what's the task? The task is to define a density profile such that the cell can read off where midplane is. And it needs to do that because at midplane it again organizes itself to form a polymer ring, and this polymer ring then switches off the E. coli cell right at midplane. So again, you see self-organized pattern formation serves the biological function, this time cell division of E. coli cells. And we'll learn about these systems and how to model them mathematically. And before we go there, I mean, if you look at those movies, I don't know whether you have seen these kind of movies, it's quite hard to imagine how you would make a theory based on these observations. Because after all, these are cells, and cells are complicated. Cells contain lots and lots of components. There's gene regulation, there's a surface, there's not just one protein, there's many, many different proteins. And the challenge really in the field is to figure out, based on these kind of data and information, of course, that you learn from people in biology, biochemistry, and all the other fields, how to describe this in terms of a theoretical model that's predictive and explanatory. Here's another example. It's a starfish oocyte, so we go up in the complexity of the organism. The starfish oocyte doesn't look like a star. This is just a tool my experimental colleagues used in order to shape the starfish oocyte in a certain form and learn from shaping it how geometry affects the self-organizing patterns that you see here. So on the left you see just the patterns. This is very disorganized, looking like spiral waves. And then this was the initial part of the movie. There's a wave running along the oocyte. And with the wave running along the oocyte, you have also the deformation of the oocyte. And that's another beautiful theoretical problem because it links self-organization of proteins in forms of patterns and shape changes, which is a domain of differential geometry. So that's a beautiful link between geometry and pattern formation. And this here is one of the latest results from Petra Schwille's group here in Munich at the Max Planck for Biochemistry. So this pattern that you see here on the bottom, this looks like a foam, like a labyrinth network in green, is actually the same proteins that make the pole-to-pole oscillation in E. coli. It's just taken out of the E. coli and put onto a two-dimensional plane. And then this object here that you see moving in this purple color is not a cell. It's not something that contains a nucleus or a machinery that would allow the cell to propel. It's just a bubble. It's called a vesicle, so it's just a lipid bilayer. And this lipid bilayer, interacting with the pattern that you see, is able to move forward just as much as you would see a cell moving forward. That's another arena of research that you see in this field. It's called synthetic cell research, and the idea is to actually be able, from the bottom up, by understanding the biological principles well enough, be able to reconstitute basic features of living systems like cell motion, cell division, etc. And that's an international effort that goes on in Europe and also in the US. Here, this is even more amazing. It's an experiment done with light sheet microscopy. So the whole field, just as a side note, has really benefited over the last 10 or 20 years by an enormous increase in ability of experimentally observing these kind of things. So microscopy, as you know, there was the Nobel Prize for that, and many other developments help to look at things at a cellular scale. So what you see here is the early lifetime of a drosophila, so a fruit fly. And what you see here is the development of the cells into a more and more organized form. I won't say much, just look at it, how these cells reorganize, form different shapes. At some point you might realize that there is segmentation going on. You have something like a midplane forming here, and here are the segments. And then you have other more complex events, and that's the clock ticking here, how long this takes from the beginning. This is in hours. Just amazing movies that see how morphogenesis happens in real time in an actual living organism. We don't quite understand how this works in detail. Understand, I mean, have a theoretical description of that. But people are getting there. People are doing research on tissue dynamics, morphogenesis, and trying to come up with large-scale theoretical models that explain something like that. And here, it's alive. So this is a quote taken from a book by Erwin Schrödinger. He wrote that when here in Germany the Nazis were in power and he had to flee out of Germany, and he wrote this book in Ireland where he gave a set of public lectures. And maybe from the movies I showed you, you can kind of learn how he came up with such a quote. He says that living matter, while of course not being outside of physics, will give you new laws or other laws of physics that were before not known. And at some point in time, these new laws will be as much part of physics and any kind of fundamental type of field theory that we have. And this is happening. So people in many different groups and labs study living systems, and they discover mesoscopic laws that are characteristic for living systems. There's more ahead, especially also with the advent of machine learning tools and other methods that help us understand these kind of systems. Interrupt me anytime you like. Sure. So now there is also complex phenomena in Munich. I think you have seen this. This is the wave. This is nonlinear physics. This is nonlinear dynamics. It's self-organized. It's pattern formation. It's based on Navier-Stokes equations, which is a beautiful equation that describes phenomena like so. Doesn't help you to surf the wave, but at least it gives you some idea of how those waves come about. This is also a beautiful picture, I find. It's clouds. It's clouds in the vicinity of the Chilean coast. It's near those kind of islands, also called the Robinson Crusoe Islands. And so those islands here shape the morphology of the clouds, and they are called van Karman streets, these kind of patterns. It's a nonlinear pattern-forming mechanism based on hydrodynamics, which is a topic that we'll talk about in this lecture. Here, this is something I guess you've seen many times, being somewhere on the beach. These are ripple patterns in shallow water. They have all kinds of different shapes. They have a certain wavelength, right? They have a certain distance. They have a stripe-like pattern or different kinds of pattern. Again, this is an arena of nonlinear dynamics and pattern formation, and there is physics-based theory to explain how granular media, which sand is, can give rise to patterns like so. Guess what this is? It's not pyramids. It's actually sand dunes at the coast of Morocco, in Morocco, I mean. And they've been there for centuries. And they are shaped by wind, and they form these beautiful patterns. It's a picture taken by an astronaut down to Earth looking at these beautiful structures. And if you ever have the chance of flying to Africa, and you're flying during the day, you can look down on the Sahara and see all these kind of beautiful structures. Right. So this is kind of to show you the beauty of the phenomena and hope you wonder how you do theory for these kind of systems. So let me give you some overview. I will do in the first part of the lecture what's called dynamic system theory. And dynamic system theory is a very general theory that's not just used in living systems or soft matter systems. It's used very broadly in all arenas of physics and also other kinds of sciences to understand systems that are non-linear. And it's following Poincaré the idea that if you want to learn about nonlinear dynamics, like the beautiful things we saw, it's not that you start writing down an equation and finding a closed analytic solution. Okay? That's not what you do. You formulate equations, you formulate theories, and then you analyze these equations, often with numerical means, and then you try to have a geometric point of view. So you look at how the system evolves in phase space, and in that phase space, there's going to be geometric structures, points, circles, manifolds, and to characterize these emerging manifolds is what you do. That's what you do when you want to understand nonlinear systems. It's a geometric point of view of phase space. And in that phase space, you want to know what's organizing that phase space, which structures are stable, which structures are attractors, and what kind of transitions between different type of structures do you get. You might know one type of phase space structure, I'm sure you know, which is statistical physics. In statistical physics, you say that on an energy manifold, the manifold actually has an equal density of points. This is the microcanonical ensemble. It's a very basic assumption about the structure of a high-dimensional system from which you construct all of thermodynamics, right? This very simple assumption that you have equal likelihood on the energy manifold is the basis of thermodynamics. And what you want to do here, once you are off thermal equilibrium into the realm of non-equilibrium systems, you would like to find a similar characterization of phase space evolution and similar laws on manifolds in this high-dimensional space. So what do you do in practice? So here are a few points. You analyze this phase space geometry. So in words, you want to know the invariant sets, cycles, manifolds, and you want to know when are they attractive and when are they repulsive. You would like to know the stability of those, and that's typically done very much so like in many other fields in physics. You look at a linear problem, so you linearize around the dynamics close to that manifold, and you determine the eigenvalues, the eigenvectors, look at the spectra, look at stability, look at the structure of these linear characterizations of the invariant manifolds. And that helps you quite a bit, usually, to get an idea of what kicks off the dynamics initially from homogeneity to some kind of non-trivial structure. What you also want to know is how do those structures that you find change. When you have heard a course on thermodynamics or advanced statistical physics, you know that there are phase transitions, right? There's transitions from a magnet to a non-magnet. There's transition from a fluid to a gas to a solid. Those are all transitions or bifurcations. And that's what you want to do here as well, not just for thermodynamic systems, but any system in general. You want to know as you change external control parameters, like temperature, concentrations, how is the geometry of the attractors, the stability of those attractors, how is that changing with these control parameters? And then also, as in many other theories, symmetry on constraints are key in understanding these systems, because they, like conservation laws or any kind of spatial or temporal symmetry, will restrict the dynamics to be on particular manifolds and also restrict the dynamics to particular type of bifurcations. All right, so that's the general theme, and now let me give you kind of an example. So here this is two differential equations. Let me focus here on the left first. just as a mathematical example, you have two variables called u and v, and they obey these two ordinary differential equations. And I bet if you look at those equations, you don't know what the outcome is, right? It's very hard to guess what the outcome is. And it's very hard to find an analytic solution. It's basically not possible to find an analytic solution. But you can still learn what the behavior of that system is in a very, very simple way. You just organize the phase space of these two variables in terms of landmark points. And there's two landmark points that should get your attention here. One landmark point is the saddle point. Right? It's an unstable point in the sense that it has only one stable direction, which is this one. And that direction is unstable. The flow, the dynamics of the system is away from that saddle. Here, that's a fixed point, because everything here in this plane, if it's to the left of this separatrix here, will flow into that fixed point. And that gives you quite a good understanding of the system, because you know when does what happen. So as a function of these parameters or initial conditions, do you flow to this fixed point or do you flow away from it? What is the final asymptotic state? And that's most of the time really what you want to know, and you don't have to know a complete analytic closed solution of the equation. You need to know the geometry of the system, right? Here, same thing. This is a famous set of equations called Brusselator, invented by people in Brussels. And this set of equations has another feature, another geometric feature that's very characteristic. It has a limit cycle. So this red line here is where all curves will end up in the long run. So independent of where you start, outside of this red circle or inside of the red circle, you will always spiral in the long run to a circle like so. These limit cycles are very important because, for example, they are the basis of circadian clocks. So your daily rhythm depends on a limit cycle that's managed in the cell by genes and proteins. All right, so that's what we want to do. We want to find these kind of attractors. And here's a famous one, the so-called Lorentz attractor, which got quite some attention in the field because it's one attractor which is not that simple, but still has a very nice and interesting structure. So what are some of the research fronts that are there? So what is actually done right now in terms of people doing research in theoretical physics on these dynamic systems? One is reaction networks. And reaction networks, I think, is very general. This can be population of bacteria. This can be population of viruses. Right? For example, COVID pandemic is one example. Analyzing how a pandemic spreads is one kind of reaction network that you can study. But also on a more molecular scale, it's gene circuits and signaling pathways in cells that are analyzed using tools of dynamic system theory. And it's via the things I told you, stability, manifolds, bifurcations, etc. You want to map out the regimes where you have coexistence of different species, where a pandemic spreads or dies off, and you want to know how to possibly control or design those kind of reaction networks so that they have properties that you want. Another theme is to turn things around, namely learning dynamical systems. So say you have some kind of data series, say the outcome of HIV infection in a person, or the outcome of the influenza infection in Germany or in the United States, or any other kind of data, and you want to capture the nonlinear behavior of that, then you often just try and learn from the data the corresponding theory. And that's a major theme in research with increasing intensity over the last years, especially because of machine learning methods. Third one is the kinetic of self-assembly. I guess all of you have some experience with IKEA or other companies where you have to assemble stuff, where you get the bits and pieces, and sometimes it's not so easy to get things assembled, and they for sure don't self-assemble, right? You have to put in some energy. But in biology, if you want to understand complex systems like the ones here, so here this is a microtubule, this is just one of those filaments in your body. This is a motor that you have on bacterial cells that drive the motion of bacteria. These are carboxysomes, and here you have virus capsids and other stuff, which is not made by viruses, but it's made by DNA origami. And Henrik Dietz, who is at the TU Munich, is one of the masters in this field. He can design all kinds of folded structures with DNA from which he can build virus capsid-like structures and other things. And if you look in the news, he has quite some big company in the meantime where he uses these insights to actually try and do some medical applications. So why is it interesting, not just from an economic point of view, understanding self-assembly? I think because it's actually a computational problem. And here I want to compare computer science and self-assembly. So in computer science, the problem is how do you solve a given complex problem with a certain type of algorithm? And there's a classification scheme, as some of you might know, of how difficult it is to solve different kinds of computational problems. And the way you would go about characterizing the difficulty is you say, you have that problem, you have an algorithm, and you have a criterion after which you're happy with the result. And then you need to know, for that kind of problem, what's the CPU time that you need to spend, meaning money and energy, CO2, given the input size of your problem. And that's what's called computational complexity, namely how the CPU time scales with the size of the program. Okay? And some are linear, some are exponential. And if it's exponential, of course, you've lost, right? Because then the time you need to solve the problem grows exponentially in the input size. When you think about assembling these complex structures like viruses and ribosomes and whatnot, you're facing a very similar problem. You want to understand the problem of self-assembly. There's a certain kind of scenario, meaning a set of rules, how things assemble into a given structure. That's the analog of the algorithm. And then you say you're happy if the structure is almost complete, say 90% or 95%, whatever you choose. Again, you have the same problem. You want to know now not what is the CPU time. For a cell, it's much more important to know what's the real time, how much will it take to actually assemble that structure, given the size of the target structure. So for example, when you think about a virus of different range of sizes, you want to know how is the time for completing the self-assembly of the virus, how is that scaling with the size of the target structure. And we call this not the computational complexity of self-assembly, but in an analogous way, the time complexity of self-assembly. Right, and the way you do this is by using methods from dynamic system theory to understand the behavior like so. Questions? I know it's an overview, it might be a bit dry. Yeah, please go ahead.
What are attractors? How do we define them?
So an attractor, I'll come to that during my lecture, but in simple terms, a fixed point is an attractor. So if you go back and look at this example here, so this point has, from here on, all of those points are in the bassin of attraction of that point. So if you start here, for example, then it will flow along that line, and this will continue attracting you towards the fixed point. So a point is an attractor. A limit cycle is another attractor. And you can have higher order manifolds that are also attractors. And this is one. This is the Lorentz attractor. So independent of initial conditions, you will land on that geometric structure. Okay? So it's a geometric structure you land on in the long run. Yeah?
How do you find those saddle points if you can't solve them?
You will learn it. This is, you look at the system, you analyze it with certain methods like linear stability analysis, and then you will see that there are certain invariant sets and how stable they are. You determine the eigenvalues, the eigenvectors, and that will then give you information about the former. So for example here, so if you're close to that point, say you can localize that point because it's one which is invariant, then you can linearize it around that point and look at dynamics linear around that point, and you'll find one eigenvalue that's positive, which will drive you away, and one eigenvalue that's negative, which drive you towards the point. So that would be a characteristic of a saddle point. Yeah? Please?
Can you consider the training of the neural networks as a self-assembling process? Because I think, for example, machine learning, we also don't have 100% correct answer. We also believe like
yeah.
to give us correct.
That's a very good and very deep question. So you're asking, given some kind of structure, like a large language model, and you train it, given data, then you find that at some point this large language model has converged to a certain structure with weights, etc. And it's a big question in the field, really a big question in the field. What characterizes these attractors, because that's what they are, and what's special about them? Nobody really knows, I would say, at the moment. This is one of the miracles of why these large language models work so well. Okay? Problem is that the space in which these models live is super high dimensional, right? And we have no intuition about high dimensional spaces. For example, in 3 dimensions, right, it's very hard to find orthogonal vectors, basically those 3. In high dimensions, every vector is orthogonal to every other vector. And that's strange, right? Or the reason why thermodynamics works is because in high dimensional spaces, all the area is on the surface. And that's the very basis of thermodynamics when you switch between microcanonical and canonical ensembles. That's another very key feature of high dimensional spaces. Okay, so let me go on. What's next? And this is not really how I'll organize it in my lecture. Just wanted to organize it in terms of themes. And one theme is phase transitions and collective phenomena. And here again, I try to put in one sentence what I mean by that. So I mean by that to have a field theory view of spatially extended systems. And a very general sense of spatially extended systems. Not just thermodynamic systems, because they are narrow subclass. Flocks of birds are a extended spatial system, or schools of fish are, or these proteins that live on the shell of a cell are spatially extended systems. And then you want to know, based on symmetry conservation laws, how you can infer from a field theory picture of that, not an agent picture, right? You want to describe it in terms of density fields or vector fields, how we can learn about instabilities, universality, and emergent macroscopic behavior, like the patterns we saw, like the flock of birds we saw. And the goal here of theory is not solving the model, it's finding the model. Okay? It's not obvious how to write down a field theory for birds. Try it. Right? It's not obvious how you would do it. Because you have to know what are the variables and what are the terms on the right-hand side that you put in order to describe all the macroscopic details that you see in the observation. So this is actually the first task. Building non-equilibrium field theories is task of theory. And that's really a lot of fun because you are then the inventor of models. You have to build them from knowing what the order parameters are, what the symmetries are, and what the conservation laws are. In equilibrium, it's fine and easy because you have a free energy, you're done, you take the free energy, that's all you need to know in order to construct the theory. Otherwise, you have to include terms that break detailed balance, meaning drive you away from equilibrium, and also break what's called Onsager symmetry. And that's a broad class of non-equilibrium field theories, and they are investigated more and more over the last years. You can also do linear stability analysis. It's now the same kind of spirit as what I talked about earlier, but you have to do linear stability analysis not just for ordinary differential equations. You have to do linear stability analysis for partial differential equations containing all kinds of differential operators like a nabla squared, rotations, just like in electrodynamics, but in a nonlinear fashion. And you need to know, based on these field theories, what are the eigenvalues, what are the dispersion relations, what are the thresholds, what are the unstable modes, etc. Again, this will give you some idea about what happens initially. So as you start from homogeneity, something gets unstable and develops. But from linear stability analysis, you will not learn the nonlinear behavior when these modes have grown to large amplitudes. And then creativity has to kick in because then you need to characterize the emergent mesoscopic structures that you find. So to give you just an example from physics you might know, superfluids are an emergent phenomena. The way to explain them is via Cooper pairs, and Cooper pairs are not a trivial extension of just electrons. You need to come up with a higher order concept. And here it's similar. If you want to understand the long-time behavior of the motion of these systems, there might be domain walls, there might be topological defects, there might be other kinds of mesoscopic structures that emerge, and you have to characterize those kind of emergent structures in terms of theory. Sometimes you can derive it from first principles by coarse-graining. Sometimes you have just to say, well, let me describe the interface as it is and try another set of phenomenological theories, just not for the particles, but for the emergent structure that you have. Let me also give you an example just to fill these words with some pictures so you have some intuition. So this here is a phase diagram, which you have learned for sure in thermodynamics. It's the phase diagram of a liquid gas or liquid-liquid mixture. Here you have the concentration, and that's the temperature. You have a critical point. Above the critical point, you have phase separation into two, in liquid and gas, or two different kinds of liquids. There's a spinodal, there's a binodal, and within the spinodal, you have spontaneous demixing. That's a well-known phenomena in thermodynamics, and when you want to construct the outcome in the long run, there's Maxwell's construction that gives you the phase equilibrium in these systems. A very old-fashioned thermodynamics. A more modern way of looking at that is in terms of equation of motion, where you say, I want to characterize the density field, which is the difference in concentration between the two components A and B, or liquid and gas. This density has to obey a conservation law, so it has to be a gradient of a current. The current is a mobility times the gradient of a chemical potential. That's basically the heart of Onsager theory. The chemical potential, from thermodynamics, is the gradient of the free energy, and the free energy is a Ginzburg-Landau functional that you can write like so. Now, you can put this on a computer and simulate it, and then you see how phase separation really works. From an initial inhomogeneous state, you have a pattern forming, but this pattern is not really stable. It coarsens over time, and the coarsening gets slower and slower, as you see. That's called Ostwald ripening. And after some time, you lose patience in the simulations. I guess it stops here, but in the end, it would completely phase separate into blue and red. Okay. So that's an example. This is another one which is more of a very modern type of experiment. It's done by Zvonimir Dodzic. Zvonimir is in Santa Barbara at the university there. And what they did is they had here a two-dimensional plane, a surfactant put on top, and then they put microtubules there. These are pretty much those objects in your cells that create cell division. And these microtubules you see here, these are these filament-shaped, elongated objects as you see them. And in between these filaments, spaghetti if you like, you have molecular motors. So here in blue, here also in blue, they are connecting these different filaments and they are pushing and pulling these filaments apart. Okay? And as they are pushing and pulling these filaments apart, you get some intriguing phenomena. You get this kind of mixing behavior in these microtubule motor mixtures. It's an interesting phenomena because it's very turbulent, very complex. And as you might see, these points here that you see, they are half-integer topological defects. So it means the dynamics of these structures is organized by topological structure in the filament system. Here are different movies, and this is an ongoing area of research. What are these active matter field theories? Well, this is kind of a class of phenomena. This we talked about. This would just be particles, not liquid mixtures as in thermodynamics, but some mixtures where the particles are actually active. And here you have different phenomena people observe. These active nematics, you find fluids which are chiral. You find metamaterials where you have edge modes similar to topological insulators where the dynamics is focused at the boundaries. And when you then analyze these different types of systems in terms of field theory, you have to write down equations for that. And I'm not meaning this for you to understand, but just to get the gist of how these equations look like. So one type of equation very much looks like the one that I just wrote down, which is the model for liquid-liquid phase separation, with the difference that this chemical potential now is not derived from a free energy. It's an object on its own. It breaks detailed balance, so it's not derivable from a free energy functional. And when you want to describe this stuff here that Zvonimir saw in his data, you need a field theory like so. And this is kind of a combination of Navier-Stokes equations and equations you know from thermodynamics. What I only want to put your attention to is that the way you describe this now is that you need vector fields corresponding to the velocity of what's going on here. And you need tensor fields because the tensors are necessary to describe the nematic structure that you have there. So this is not just density, it has orientation. And to capture orientation, you need tensor fields. So these are tensor-like field theories that one has to derive and analyze with the methods that I just explained. Some research frontiers. One is this field of active matter. This comprises quite a few different systems. Starts from self-propelled colloids, populations that interact by chemical fields, or active emulsions where you have particles that driven by energy from, say, ATP hydrolysis are driven far from equilibria. And what nonlinear dynamics does is via all the tools that I told you, stability analysis, field theory, interface theories, maps again out which regimes in the system occur, and how they shift as a function of parameters. So in the end, it's generalizing the theory of phase transitions, because in phase transitions you want to know how a control parameter changes from a fluid to a gas or from a paramagnet to a ferromagnet. Here you want to know how control parameters change the behavior from simple to more complex to even more complex kind of behavior. Another one is very much in biology. In biology, people worry a lot about what's called biomolecular condensates. These are structures within the cells that are different from organelles. They are called nuclei, stress granules, or transcriptional hubs. And those are new objects in the biological organization of the cells that are not thermodynamic objects, but again driven by nonlinear reactions and input of energy. And there you would like to know how different regimes where these condensates nucleate or grow, how they organize in different structures, position themselves, oscillate, wet the structure, divide, and whatnot, how this all occurs and how this depends on the energy you supply, on the kinetics, and how a cell with the genes and all of it in it is able to regulate that. And another one also, similar as I mentioned before, people would like to know how you can actually infer just from data theories and identify order parameters and even equation of motion by learning them from a given set of data. Questions? Yeah?
The equation that you gave us about the active non-equilibrium field theories,
Yeah.
it's the same for every kind of system that you presented to us? Or
There's lots of different field theories. What they share is sometimes that the variables are the same. So say you have a field theory that's containing a scalar, a vector, and a tensor, or higher order tensor fields. That's pretty much universal because that's what you observe in terms of the morphology of these kind of systems. When it comes to the right-hand side, it's very different because on the right-hand side you encode all the interactions that you have in these systems. And there, depending on the system you look at, you will get different terms, either structurally different terms, so meaning different kinds of gradients and powers of the fields and couplings between density and vector fields. And for sure, of course, different parameters, but so both. Parameters may change and the structure of the theories change. Over the years, there have been certain classical models that have been, coming out and being characterized. So you have a classification, more or less, of the type of models that you get. Yeah. Okay, it's just an overview, right? This is for you to give you an idea of where things are going, to whet your appetite, so to say. And we do theoretical physics here, right? That's what we do. Now, last, pattern formation and self-organization. This focuses more on one of the most beautiful complex systems that we know of, which is living systems. And it tries to have a unified view of these living systems in terms of their reactions, their transport, the active cytoskeleton. So you saw this folding and unfolding of the microtubules. That's an example of a cytoskeleton of a cell. And you want to understand how is this shaped by the symmetries of the problem, the conservation laws, and again, the geometry of the system that you're in. So you saw this starfish oocyte. Remember, it was deforming in space, and you would also like to understand how geometry affects that. And then you want to know what kind of patterns evolve. Is there this polar cap that's responsible for budding in yeast? Are there these oscillations back and forth in the coli cell? Or are there more complex patterns, like maybe patterns that underlie the beautiful morphogenesis of the drosophila egg that I showed you at the very beginning? Right? How do patterns maybe guide the formation and reformation, the reorganization of tissue in a living organism? And that's kind of an agenda that's out there worldwide in different groups and different labs to understand living systems from this perspective. What we will do in the class is we'll start from the classics. There's quite a range of classic models in pattern-forming systems. They run under various famous names, starting with the work of Alan Turing. So Alan Turing in 1952 wrote a very influential paper where he started to think about morphogenesis in terms of diffusion reaction equations and came up with what is nowadays known as Turing patterns. So he not only came up with Turing machines, but he also was responsible for quite a paradigm shift in studying biological phenomena in terms of diffusion reaction systems. And then there's other names, Swift and Hohenberg made lots of contribution, Ginzburg and Landa, and Kuramoto and Shibashinsky. They developed a range of different classes of mathematical models that one needs to kind of know to understand the basics of pattern formation. And the way you analyze or will learn to analyze those systems will be through the analysis of dispersion relations using linear analysis, spectral analysis, and then from there go and derive equations that are valid deeper in the nonlinear regime called amplitude reductions, and they will hopefully then allow us to predict features of those patterns, wavelength, wave period, when they become stable or when they are unstable. One particularly important class that we'll look at are so-called mass-conserving reaction diffusion systems. That's important in biology. In biology, it's very difficult when you look at these patterns on a yeast cell or in E. coli to produce or degrade proteins on the time scale at which you see the patterns. Proteins are produced at a time scale of many, many minutes to hours. The patterns happen on time scales of below a minute. So you don't have loss or creation of new stuff. And that's encoded in mass conservation in those systems. And that's essential because mass conservation puts a constraint, and these constraints often are highly relevant in understanding how the behavior of a system changes. So that's what we'll do. We'll use conservation laws and flux balance geometry in phase space to look at a geometric classification of patterns in the, or pattern instabilities in these systems. And again, there's this step from learning about linear instability to understanding the emergent mesoscopic phenomena, where you see interfaces, waves, and all kinds of other patterns. And there we will learn how to construct theories for these mesoscopic patterns, like the interfaces or forms that emerge in the nonlinear regime. Again, I want to illustrate these dry words by some examples. That's the famous Swift-Hohenberg equation. So it looks very harmless, right? What you have here is something like a 5 to the 4th theory that you would also have in thermodynamics for ferromagnets. What's special here is that you have this differential operator, which is not just a nabla squared, but a nabla to the 4th, and combined with a certain wavelength QC, which you can basically think of is the basic length scale that determines the pattern. And when you study these equations numerically, you'll see they form patterns like so. If you adjust the parameter mu to 1, of course in dimensionless units, and QC to 1, that's a pattern you can get. Or if you adjust it to some other values, you no longer get stripe and labyrinth type of patterns, but you get a crystal-like structure where the dots that you see here are arranged in kind of a hexagonal type of pattern shape. Okay? And we'll learn in the lecture of why one pattern is formed versus another pattern is formed, and what's the transition between the two, and how do we understand and learn about this. I showed you that. How am I doing in time? Yeah, fine. But interrupt me if you have questions, otherwise I'm too fast. No? So here this is this E. coli cell that does this oscillation back and forth between the two planes, and you have to think about this as a spherocylinder, right? This is not a a plane, there's a spherocylinder, and the pattern is on the surface of the spherocylinder. Inside the E. coli cell, you don't see the pattern. It's really on the membrane, on the surface of that. And it goes back and forth. Time scale is a minute. And as said, it creates this density profile that localizes things at midplane. And what's amazing is that just 2 proteins do the job. They're called min-D and min-E. That's all you need in order to create that pattern. How much of you know the B-set reaction? None? Chemistry, you know? Okay, so there's chemistry, and people have found at some point in time that chemical systems show pattern formation. And that was a surprise, because people didn't really believe that these chemical systems would be able to show pattern formation. So B-set reaction was a very, very important step in the development of the field, and it won many, many awards when people found that out. The strange thing about pattern formation in chemistry is the B-set reaction has something like 1,000 components. Okay, it's very hard to wrap your head around how to understand and analyze these kind of systems. The protein system that you see here, the min system, just has 2 proteins that do the job. It's quite amazing that 2 proteins can do that. So how does that work? So just to give you the gist of it, there's a membrane, there's this spherocylinder on which the proteins live, and there's the cytosol. And in this organization of the pattern, there's 2 key elements, and they're both very much physics-based. The first element that you have is that you have the system being driven by an NTPase. Many of you won't know what this is. It means energy input through ATP hydrolysis in your body. Every one of you burns 30 kilogram ATP a day. Okay? You don't lose weight by that because it's just a turnover of ATP in your body, but that's how much energy you need in order to run things in your body. It's the basic energy unit that drives all living organisms. And what it does here, it changes this min protein from an inactive state with a diphosphate, the inactive state, to an active state with a triphosphate bound to it. Okay, and that's where energy locally flows into the system. That's where you kick the system off thermodynamic equilibrium, and that's where thermodynamic loses its validity right here in this biological system, because you push the system out of thermodynamic equilibrium by feeding in energy through ATP hydrolysis. So what does that achieve? So you feed in energy, and as you feed in energy, this object now, the activated form of the min proteins, is able to bind to the membrane. The other inactive one is not able to bind. Okay? So activation changes the affinity of the protein to the membrane, and then it switches, or it transfers from a 3-dimensional bulk, the cytosol, to a 2-dimensional manifold, the surface, the membrane. So once it's there, it kicks on a nonlinear feedback. It recruits more and more of the min proteins to the membrane. Okay, so if there's a density locally at the membrane and something comes close, there's some kind of interaction that pulls more and more min proteins to the membrane. So we have an accumulation of proteins, of these min proteins on the membrane. That would not do the job, right? Because then we just flush the membrane with stuff, but you wouldn't get it off anymore. So what you need is an antagonist, another protein that's called min-E. It's also recruited to the membrane by the min-D, so some kind of interaction. Then they make a dimer, and in this dimeric form, this min-E protein switches the min-D off. So it turns it from this active form back to the inactive form. And now it's inactive, it can no longer hold onto the membrane. Both fall off, and they're back in the cytosol, and this whole cycle here starts again. Okay? And that's a hallmark of non-equilibrium. You have a non-equilibrium flux of proteins between different states and between different compartments. In thermodynamic equilibrium, you have no fluxes whatsoever. Right? So this is really because you drive the system by an NTPase. And that is a major theme why these systems show patterns at all. If you turn off the ATP hydrolysis, pattern is gone. Mathematically, this is how it looks like. So what you have to solve is diffusion reaction equations. So you have some vector of components that diffuse. There's some reactions on the two-dimensional manifold. There's a vector of components in the three-dimensional volume that diffuse and show some reactions. Everything conserves mass, and you have a boundary condition between the membrane and the cytosol, and that boundary condition balances the reactions that you have with any kind of diffusive fluxes to or from the membrane. So it's a coupled set of partial differential equations with boundary condition. That's the mathematical problem that you need to solve, just to show you the structure of what's going on. And as I said, it becomes even more beautiful if this manifold here is not a constant manifold, but changes its structure, its geometry, the curvature, and then you have an interesting intermingling between the changing geometry of the boundary and the diffusion reaction system that you have. Okay? And by the way, these diffusion reaction systems, they read off the geometry. Like, you know the question, can you hear the shape of a drum, right? The answer is yes, because the boundary determines the eigenmodes. Here, it's not a linear system, it's a nonlinear system, but still that boundary here and the coupling to the bulk makes the system be able to sense the geometry and therefore read off the geometry. And thereby you have this interesting coupling between the geometry affecting the pattern and the pattern by attaching to the membrane, changing the geometry of the manifold. This is how it goes, right? Here, this is this example I showed in the beginning. You have the pattern couples to this manifold, and when the movie starts again, you see how from the beginning there's this wave running along the periphery of the oocyte, and this wave carry with it a geometric deformation. So pattern formation and differential geometry, quite a challenging theoretical task. So there's more open research questions, and I close with that. One is, it's kind of a combination of Turing 1 and Turing 2, because Turing came up with Turing machines, and Turing came up with Turing patterns. So question is, can you compute with patterns? The answer is yes, because biology does that. It computes on the basis of a given pattern, organization of tissue and proteins and gene regulatory factors. The next stage, that's the very heart of morphogenesis. And Turing put this very nicely in his 1952 paper. He said the actual challenge we have to face in understanding biology is this one, namely that most of the time organisms, they are developing from one pattern into another and not from a homogeneous steady state into one pattern. And this going from one pattern to the next, to the next, to the next is a computational problem in space, just not formulated in terms of Fortran or C++ or any other kind of language, but in the language of biology, namely the language of proteins, their structure in space and in time. Reactions induce phase separation. That's another frontier in this field, namely whether when you combine chemical reactions in a cell and phase separation in a cell, can you combine the two to actually get even more intriguing phenomena that explain some of the biology that happens in cells. Last but not least, can you transport stuff with patterns? I showed you an example. There was this vesicle, this bag of empty stuff moving on a pattern. You can go even further. You can actually move cargo with chemical reactions, and this is one of the cover pictures of Nature Physics, where the experiments of Beatrice are shown, where he showed that these patterns that I alluded to can actually push things around, and that could be another very interesting feature that organizes biological matter.